As in the other webinars in this series, we'll go through Kat's academic and work experience to find out how she got to where she is now, and to use this as a general conversation starter about entrepreneurship in the environment. If you have any questions for Kat, please put them into the chat box and I'll try and weave them into our conversation. So I'm going to hand over to Kat, who's going to give us a little bit of background of how she got to where she is today.
Cool. So I would guess the best description of my path to where I am today is non-linear. I started off studying, well, I did A-levels in classical subjects, so Latin, ancient Greek, maths and French. I'd had a moment when I was deciding what A-levels to do where I sort of thought about going down the sciences route, and then I don't know what tempted me towards ancient Greek instead, but something did. I like the stories. And that sort of led naturally into a degree in classics at Oxford University, where I was studying, having to read the whole of the Iliad in ancient Greek and translating Shakespeare into Latin and all sorts of weird and wonderful things. And at some point during that, I suddenly had a bit of a revelation that I guess almost by definition, classics is not the future. And it wasn't really where I felt that I wanted to be. So I somewhat unceremoniously and without a lot of warning dropped out of the course at Oxford. I had a brief period of threatening to go and become a hairdresser in Peterborough that my parents weren't too keen on. And eventually I was working in a pub and the manager said, "Oh, why don't you just go feed lions in Africa or something?" So I think I literally went home and Googled feed lions in Africa and came up with this random expedition that was heading off into the Amazon rainforest in Ecuador in about a month's time. Threw in an application and they were full, but managed to squeeze me in.
So as a teenager, had you been interested in the environment, or was this a university conversion?
I was trying to run away mainly, but actually when I look back, both of my grandparents were professors of geography. My grandfather studied deserts and climate, and my grandmother was a glaciologist. My uncle was an environmental historian. My dad's a zoologist. So in a way, it's kind of fate coming back round to get me. I tried to get away from it, but yeah, there was something in the genes that led me back to the Amazon. So yeah, I found myself in the middle of the Amazon, sort of 100 miles from the nearest road, with really no idea where on earth in the world I was, or why I was there, or what the Amazon was. But after a while, I discovered it was pretty good. And weirdly, I think the skills that I'd learned from the classics, which included, I did a lot of things like doing ancient Greek tragedies as performances, where you'd have to learn sort of 20 pages of ancient Greek verse by rote. You didn't know what most of it meant, but I think I had a really good ear for sound. And so I turned out to be really good at learning bird calls. So I got really involved in the research in the Amazon because I was able to identify all of the birds by their calls. And that kind of made me think, oh, maybe this is what I want to be doing. So I spent a couple of years going back and forth to the Amazon, leading expeditions and working in research centres and things, and was eventually persuaded by somebody very wise that I ought to go and get a degree.
Which part of the Amazon were you in?
So that was Ecuador and Peru. So on the sort of western side of the Amazon. I learned Spanish out there, so I had a weird vocabulary to start with where I could discuss at length about whether the firewood was good for burning, but I had no idea how to book a hotel. So I went back and did an undergrad at Anglia Ruskin University in Cambridge, which I have to say I loved. As a sort of, I'd been at Oxford and sort of seen what that end of things was like, and it was so refreshing being surrounded by mature students who were all bringing something different to the table at Anglia Ruskin. We did a lot of very practical work, a lot of field work, scuba diving, and I had a lot of really inspirational lecturers there. So then I was linear for a bit, and I sort of went straight on from that to masters at Imperial, where I did entomology because I felt like I'd missed out all of the small things in my degree, and that was where I first had to go doing some genetics. So my first project there was sort of a phylogenetic project on wasps. I didn't really like it very much, and I wasn't very good at it. So something deep inside me doesn't accept that. So I thought I'd better go and do a PhD in genetics just so that I could get over that dislike of it. So I and my PhD that I got at UEA sort of tied in back with my tropical experience, and it was doing population genetics of an ant plant system in the Peruvian Amazon. So I did that, spent some time in Peru collecting ants, spent a lot of time sitting in the lab pipetting tiny amounts of colourless liquid into other tiny amounts of colourless liquid, and wondering whether I'd got my plate back to front. And after about halfway through my PhD, I thought, well, I was qualified for was sitting on a factory production line, and lost it, sort of started wondering why anybody was interested in how far these ants moved anyway. And at that point, my PhD supervisor was just getting interested in this sort of new approach where you could take, you know, a whole unsorted sort of bulk trap sample of insects, and instead of having to look at them one by one under the microscope, you could just blend them all up and make a soup out of them and extract all of the DNA and sequence it.
Right.
And you could do that for sort of 100 samples together at once and get these huge data sets that gave, you know, biodiversity data at scales we'd never seen before. And sort of thought there must be something in this that's useful for environmental managers to be able to sort of track how habitats and ecosystems are changing, either sort of through natural change or as a result of management or impacts. So we found that actually those tools were all being really well road tested by academics, and then they would be written up as papers, and the last paragraph always said, you know, this is going to solve so many challenges for environmental managers, it's going to be the best thing since sliced bread. And they published it in molecular journals, and the environmental managers never heard about it. Mm-hmm. So that was a sort of obvious gap, and that was where we thought, okay, there's a real sort of opportunity, not only as a business opportunity, but also it would be unbelievably frustrating if we are sitting here knowing we can do all of this, and the people who it would help don't know about it. So that was where we came up with the idea of starting a company.
Okay, so to summarise your academic career, you're a classical environmental genetic ecologist.
Tropical ecologist,
okay.
Yeah. Pretty much.
Yeah, so that's a key thing that keeps coming through in all of these entrepreneurial webinars, is that translation between research and the real world, the kind of research and applied science. And I think the university sector is well aware of that gap, and things like REF and impact case studies are really seeking to kind of address that, along with a lot of funding bodies. So that might be something that we come back a little bit to later on, about how your experience might kind of encourage the formation of those relationships. So start us off with your first startup.
Well, so I originally went and got an internship actually at a startup accelerator called Wyra, which was an interesting place. It's run by Telefonica, who own O2 and companies like that. And a lot of these big companies are realizing they're not agile enough to innovate very well in-house, so they're instead bringing in lots of startups and giving them skills and investing a bit, so they have a bit of a stake in them, and innovating that way. So I went in to help run the accelerator and sort of assist with the program, which was fascinating because I could look across sort of 25 at the time different startups and see, you know, what are the things that everybody is doing the same, what are the common mistakes, what are the different sort of options and ways of going about things.
Geographically, where was this based?
In central London. It was on Tottenham Court Road. So it's a really cool sort of vibrant space that we were in, and kind of quite sort of open working, and everybody, you know, I think really key, everybody going through that same journey together, and a lot of sort of peer support available and linking to networks. And was
Telefonica a funding partner in that, or were they just providing the space?
Yeah, so Telefonica sort of, you know, they invested money in each of the startups, and were sort of quite involved, but there was some of the startups had a direct link to something that was clearly sort of central to things Telefonica were interested in, others were a little sort of less.
And your idea being in that category or not?
So that wasn't anything to do with NatureMetrics.
Okay.
I went there because I thought, I want to, you know, I'm interested in this idea of starting a business. I think we can see that there's something interesting to do. I don't have first clue about businesses or startup businesses or where to start or what any of this stuff is. So this was just completely separate to go and learn a bit about startups. And then I found after a few months I was recruited to go and work for one of the startups that was in the accelerator, which was a social impact startup called Charisma Kids, which was about as far away from my idea of squishing up ants as you can get. So this was making sort of computer games for young children to help them learn emotional intelligence. There was this whole sort of set of characters that would teach children about emotions and things like that. But obviously at the same time we're sort of building a business plan and pitching to investors, and sort of so just getting an idea of what those processes are and being part of it was an amazing education for me. And then we got a little bit of funding that would support me for a while to go and work on seeing if we could get NatureMetrics set up and running. Mm-hmm. And that was at the beginning of 2015. So from that point on, I've been sort of full-time on NatureMetrics. And just to give you a bit of a sort of an overview, I guess, of what it is that we do in NatureMetrics, we specialize in using DNA from complex samples to be able to survey species. So that can be quite a lot of the time at the moment it's a water sample. So you can take a water sample and anything that's sort of moved through the water recently within the last sort of day or two leaves a genetic signature in the water. So it's basically like a sort of soup of all of the wildlife that's been present within the last couple of days. And you can just filter that water and track all of the DNA on the filter and then sequence the DNA to identify which species were present. And you can do exactly the same approach with the insects, which is where I started. So you can set up the traps and collect thousands of insects and then blend them and extract the DNA and sequence it in the same way. And we're just at a point now where, for example, we went out a couple of weeks ago or a couple of months ago with some volunteers at a BioBlitz event where they never include fish because it's too difficult to do. So we took some volunteers out and they collected some water samples and we sequenced them. And in one sample collected by a volunteer, they found that every fish species that the Environment Agency had found at that point since 1990, plus an endangered species that hadn't been found by them. So it's pretty cool what we can do, but it's a new market. So it's been very much a sort of educational process in working with potential clients and sort of helping them understand what these tools are and what they can do and what they can't do, sort of how to be intelligent users of them.
So are you the supplier of the equipment, the people, and the genetic database of species, or is that database somewhere else?
So we mostly start with sort of public universal reference databases, but the sort of most extensive reference database is also the least well curated one. So we do quite a lot of our own sort of curation of those reference databases to get rid of the errors and make sure that what we're using is good quality stuff. Because you have quite a lot of instances where people they think they've sequenced their study organism, but they've actually sequenced themselves. So, right. So there's all these human sequences masquerading as some insect, and it causes all kinds of problems when you go and do taxonomic assignments. So we have to try and get rid of those problems. And actually in the UK it's relatively straightforward. Once you start going to parts of the world where the diversity is just much less known and there's very little in the reference database, then it becomes much more complex.
So those reference databases open source, or do you?
Mm-hmm.
Okay.
Yeah. Quite often, especially if we're going to places where we haven't done work before and there's big gaps, then they can be supplemented with collecting. If you're doing fish, you can collect fin clips from the fish that you catch during that first survey season and fill in some of the gaps.
So we've had our first question. Someone's asked, I suppose it's not possible to know what the organisms have been doing, living there, visiting, eating or defecating.
No, you can't tell so much what they've been doing. Interestingly though, behaviour does affect the detectability. So if you've got organisms that are very active and moving around a lot, and they've got their metabolism's working and they're excreting a lot, then they put a ton more DNA into the water than if they're just sort of sitting there still at the bottom. So it does come into play, but we can't tell from the data.
And Guy, I think, as a follow-up to that, says, what about stomach and fecal bacteria?
So you can do, so basically once we've extracted the DNA, the nice thing about this approach is that once you've extracted the DNA, whether it's from water or feces or stomach or whatever, you've got the DNA of everything there. And you can then choose, in most cases, we're interested in the, vertebrates or invertebrate animals. And so we have a particular amplification process that amplifies the DNA of that group that we're looking at. But if afterwards you go, actually, it'd be really interesting to know what bacteria were there as well, you can go back to that same DNA sample and amplify the bacterial DNA and sequence that as well. So we haven't done a whole lot of that, but it's absolutely something you can do.
Excellent. Okay, so I was just waiting for the question to come through there, but he stopped typing. So if we move on to how the company has developed.
So we started off in the beginning of 2015, really, and what kickstarted everything was some funding that UEA gave us, which is where I did my PhD. And for them, they've been an absolutely key partner for us. And for them, we're a really big impact story. And so what you sort of were alluding to before with the ref and things, that really played into our hands, and they've been incredibly supportive. So they gave us a small grant, which basically sort of funded me to be able to work on developing the business plan and finding some more money. And then shortly after that, we got a NERC follow-on fund, so grant. So I was a NERC PhD student, and NERC provides some funding for sort of building on PhD studentships and taking that work sort of more towards application or commercial viability.
And in those funds and that you get from UEA and NERC, are they restricted funding? And are they taking a proportion of your company for that?
So in these particular cases, with UEA, kind of. So they did end up. It wasn't at the time. The deal wasn't kind of we'll give you this much money for this much equity, but they ended up having a stake in the company for the support that they'd given us in the early days, and they still do. With NERC, no, NERC funding didn't necessarily have to lead to the formation of a company, and they wouldn't take that was sort of pure grant funding. We had and that was sort of allowed us to run I think 3 different pilot projects. So with real potential clients, but doing it through the universities. So it did mean that we had to pay a lot of university overhead. So that number of 250k looks really high, but a lot of it disappeared straight away in university overhead. So we basically had myself and another postdoc for a year working on these projects and getting some data. And during that time, we also ran our first commercial service, which we did again this time through Imperial College. So my 2 co-founders, one was a professor from UEA, Doug Yu, and the other professor at Imperial, Alfred Boker. And so through Alfred's lab and Imperial consultants, we ran our first service, which was testing water samples for great crested newt DNA, which I think this was the second year that test had been approved. So it actually got the regulators and government were very, very quick to adopt DNA testing for presence or absence of great crested newts. So they sort of had full approval, and the people were already asking for it. So it's kind of low-hanging fruit in terms of something that you can do and generate some revenue straight away. That was a really nice story to be able to put into an investment pitch and say, look, we've already been able to run a service, we've got some customers, we've done, delivered good quality of service, they want to come back to us. You can see there's some money to be made here. So
in these early stages, did you do any comparative with more traditional forms of ecological sampling, so baits and traps and that type of thing, to see how your system performs against theirs?
Yeah, so we did quite a lot of that actually. So actually going back to my PhD, so we would have, for example, we piggybacked on several projects where they'd already done, say, pitfall trapping. There was one project in particular looking at heathland restoration, where they had monitored the heath they were trying to create, and then monitoring the trackways they were trying to convert. And they'd already identified everything, and we just went along and took all of their samples and squished them up and sequenced them. Basically replicated somebody's PhD in about 6 weeks. Which was slightly embarrassing.
You didn't tell the poor individual,
no,
did you?
Yeah, we worked with him on it. But what was interesting in that was that we weren't necessarily looking at, you've got 2 data sets. Do you find exactly the same species in both data sets? But we were saying, when you analyse the data on using community analyses, do you see the same trends and the same patterns? Is it telling you the same thing about how the environment's changing, and would you make the same management decisions based on them? And across the board, the answer is yes, you would, and you get more statistically powerful data, and you can do a lot more of it.
Okay.
Yeah.
So there'll be some ecologists who spent a long time doing kind of species identification training, knocking at your door, or pitchforks.
Well, it's been interesting actually. We expected a lot more antipathy from traditional taxonomists than we've actually had. I think back in the day when the concept of DNA barcoding came up, I think it was a lot more vicious. I mean, if you read some of the papers in 2003 when the idea of identifying things from DNA barcodes was first put forward, I mean, they are really vicious papers going back and forth. And now actually people are kind of okay with that idea. And actually, the reality is that we have to work with taxonomists. We need taxonomists to be able to generate those high quality reference databases and fill in all of the gaps and validate what we're doing and all of that kind of thing. So I think, a lot of people are seeing it as an opportunity.
Okay. So related to a question that came in, how careful did you have to be about defending your IP in those early days?
Well, that's a really interesting question, and it's still an interesting question today. Fundamentally, what we were doing was taking published research and applying it commercially. So we didn't actually start off at a point of having a piece of IP that we were commercializing. And we went through this whole process with Imperial, who was sort of saying, oh, well, you need to be an official spin out and sort of write down exactly what your IP is. And we sort of tried to explain that we didn't really have any, and they didn't really believe us. We got right through the end of the process, and they said, point out the thing here that you can do that nobody else can do. We said, there isn't anything. They said, oh, you haven't got any IP then? So just sort of go and start your own company. So no, actually, we haven't been massively IP focused, and it is something that we're now sort of having to think about in terms of, as we grow the business, what do we have?
Okay, so if we continue back with the story about how you did go about growing the business.
So big moment for us was beginning of 2016, where we managed to raise 250k, which was mostly from business angels, so private individuals investing their money. Mostly they're quite experienced investors, so they know what they do. They have sort of a portfolio of companies that they invest in. And we had a group of them that they each put in sort of a relatively small amount.
So how did you go about finding those individuals?
So I used some of the routes that I'd learned about through working with the startup accelerators and the startup before. There are organisations that sort of bring together groups of investors and then go out and find interesting startups to bring and put in front of them. So I found my investors through 2 of those networks. There was the Angel Investment Network and London Business Angels. And you have to sort of go and chat to the organisers, and then they decide whether they want to bring you into one of their pitch events, which is sort of an evening event with maybe 100 investors in the room, and you sort of stand up and have 10 minutes to explain everything about your business.
And did you get the sense that those investors had a specific science interest, or were they looking across the board at all different types of companies?
A real mixture. So I had one of my investors, who's now the chairman of our company, he had a real sort of interest and passion in conservation and had a sort of a long history in green investments. So that was kind of a really natural fit. The other investor who's ended up on our board, really his interest is in data. And then I've got somebody in there who's invested previously in some genetics startups, and some of them who just see it as an interesting business. It's a challenge when you have these things that, you literally have 10 minutes to get up on stage, and if your business is basically, oh, we create an app to help you find something when, everyone knows it's difficult to find, then it's a pretty easy story. But for me, I'm having to get up and explain, what is biodiversity? Why do we care about biodiversity? Why is there any money in it? Who on earth would pay for it? As well as like the whole business plan. But I think in some ways, because it's something a bit different from what people were seeing all the time, I think it sort of got their interest.
Okay, we tend to talk about the science biotech clusters being around places like Cambridge, but I've noticed a lot, you've talked about London. Is there a cluster in London as well?
There are. There are biotech incubators at Queen Mary's and the vet school and places like that, and there's ones at Stevenage and Welwyn Garden City, and all these places that have got lab space that you can go. When I first went around looking for lab space the year before we got our funding, there was space available in just about all of them. A year later, I went around and the whole of London was full. The whole of Stevenage, Welwyn Garden City was full. There were a couple of new ones being built that were already pre-booked. Cambridge was full, so the closest place would have been Norwich.
And what's the explanation for that?
I think the sort of biotech sector is just, in a real sort of boom phase. I think there's a lot of interesting companies. Most of them are sort of more on the biomed side of things rather than environmental. So we were lucky that I met the director of CABI, which is the Commonwealth Agriculture and Biosciences Institute International, and they're based in Egham in Surrey, and they do a lot of work on sort of agriculture and capacity building worldwide. And they had really good molecular facilities, and they just bought a high throughput sequencer, and so it seemed like a very natural place for us to go, and we would be able to sort of support them with their work, with their sequencing, and sort of vice versa. So we've ended up setting up there.
So you got this 250,000 in equity funding. What did you spend it on?
A lot of it was setting up the lab. So there's obviously quite high capital costs in doing that. It was amazing that we were able to go and live with CABI and sort of have access to some of their bigger equipment, so we didn't need to buy all of that. But yeah, that was a big cost, and then employing people for the first time. So I sort of had 3 staff, started pretty much right away, those are the main things really. We haven't done a lot of marketing or anything, because we didn't really need to at the time.
Okay, so very high upfront capital cost. Does the equipment depreciate very quickly, or is that an investment for the long term?
It's somewhere in the middle. Some of the equipment we use, on a daily basis, will not have a massively long life cycle. So one of the things is that between April and June is great-crested newt season. So during that period, we have huge numbers of samples coming in, just looking for great-crested newts. And we have to follow this protocol that Natural England sort of set out, which is based on centrifuging tubes of ethanol at really high speeds. So we had to invest in, a lot of these really massive centrifuges that could spin those sorts of samples at that speed, and we only use them in that 3-month period. So yeah, that's probably one of the costs that we wish we hadn't had to.
Although if you say that you don't have a huge amount of IP, very large capital cost is quite a good way of preventing too many competitors entering the market. So it could be useful for you. Yeah,
there's that, and it's just that, if you go end to end of the process of doing high throughput sequencing on an environmental sample, there's probably 150 different steps in there. And the computational side of the data analysis is also, you can do everything right and get great raw data, and then you can get meaningless information out of the end of it if you don't process that data right. So a lot of our expertise and know-how and sort of IP is in how you do that data processing in an automated way to give meaningful data.
Okay.
And then very quickly after we set up, we almost destroyed everything by contaminating our labs with great crested newt DNA, which is one of the massive risks of what we do, because you're working with tiny, tiny amounts of DNA.
Yeah. I remember something similar happening to the government forensic labs when they privatised those, and then they found about 3 or 4 years of data, I think it was, had all been contaminated, and you imagine that all tied in with the court system and the expense around that.
Yeah. So this was right at the beginning of newt season, and you're suddenly in a situation where your negative controls are all coming up with newt in them, and having done this great job of sort of setting everything up and building this network of customers and clients and lots of orders, suddenly we were in a bit of crisis mode, and for me that's sort of, I'm feeling like, oh my goodness, I've just taken everybody's money and I've employed people and I've got all these responsibilities, and now I'm going to let everybody down straight away. And so it was kind of a massive learning curve about how you deal with those situations, and we were really upfront with people whose samples it affected, and we found a way to sort things out for them, and sort of managed to get through, and then use that as a learning experience and put in place, sort of 10 levels of security to make sure that would never happen again, and set up a whole separate lab at a completely different site where we only extract DNA from water samples. And so, we sort of decided at that point, okay, we're going to make sure that we are absolutely 100% the most robust lab out there, and we're never going to get contaminated again.
And were your business angels, investors, sympathetic during that period?
They were. I was lucky that the people who had invested were experienced. They've seen everything before, and that's actually one of the sort of big take-home messages for me, was that the investors are about so much more than just the money. These are experienced people who, you feel like everything's crashing and burning, and for them it's just another startup having a bit of a difficult patch. So they were really, really supportive and helped me sort of find my way through that situation. And actually it wasn't a very long period of time, but it just for that period it felt like the world was collapsing a little bit.
So the kind of opposite of the Tesla experience that is publicly listed, where they feel that the investors have got too short-term an attitude around everything, your investors were able to look a bit longer into the longer term.
Yeah.
An argument for staying private.
Yeah, and they, I think so. I just, I felt very, very lucky to have those people on board with me. So that at the end of that year, we set up our second site, with sort of positive pressure clean rooms with UV lights, and nothing other than extracting DNA from water samples on that whole site. And then I was trying to think what we've done since then. Basically, we've just been slowly building our offering, sort of honing our techniques and getting better and better at what we do. Educating the market and the clients on, you can do more with environmental DNA than just look for great crested newts, and building up the team, learning something new literally every day. And I mean, I've got involved in things during that period. Like there's a big European group called DNAquanet, which is a cost action program from the EU, where they give you lots and lots of money for 5 years to not do any research but to talk to each other. And it came at just the right time for this field, where actually people have been going off on their own tangents, all doing different things in the same space, and we needed to come together and talk to each other about what worked and what didn't work. So I'm leading a working group in that to sort of try and find best practice methods in the field and the lab. So that's been a really good experience. And then just recently we've suddenly just started to feel like we're a little bit more established, and we should make a small profit this year, which would be really exciting. So that's our first year of being profitable, and things are feeling more stable now than they have done
And how many staff are you up to these days?
We've got 10.
10 staff, yeah.
Yeah. So.
And how many customers?
So I think my next slide shows that. So yeah, we've got 10 staff and I think about 110 different customers. We range from sort of, environmental consultancies, some very specialist ones, some very general ones, and then sort of conservation organisations, and some universities and research groups as well. And we've had samples from all over the world.
Okay, so what are the barriers to the widespread adoption of this approach?
Partly it's that it's new and people don't know it, so people are wary to try something new, and that goes through. For us, it's very easy to talk to environmental consultancies, but then actually the environmental consultancies have to go and sell it to their end user clients. So what we've realised recently is that our sort of education process, I guess, has to include those end users as well as the consultancies, so that there's a sort of continuous stream of understanding. And then you've got things like there's obviously some situations where you need to count things, and DNA doesn't count things very well. It can tell you whether something is there, and it can give you an idea of the sort of relative abundance of the different species. But if you need count data, or if you need age class or size class data, then you still need to go and catch things and look at them. And then you've got we've been trying to be very clear and open about the limitation. But it's funny because people get scared by that, whereas actually everything that people are used to using has its own sets of limitations and biases and things, but people just don't talk about it. So yeah. Is
there any regulatory barriers? Is it written into guidance? And
there's some. It varies depending on the context. So, for example, it's a really great tool for analysing communities of freshwater macroinvertebrates. You need to collect the macroinvertebrates and squish them up. You can't do it very easily from the water, but you can get great data. But the Water Framework Directive has the word abundance in it, and therefore we can't replicate that abundance information that's needed for Water Framework Directive monitoring. So that kind of means that actually that's not a very big opportunity for us at the moment because of that regulatory barrier. So part of the EU project that we're working on is about sort of preparing these techniques for being integrated into Water Framework Directive monitoring and looking at whether we need new indices and new approaches and changes to the actual directive. But it takes a very long time to get those sorts of changes.
And what kind of mediums can you sample in? So you mentioned water bodies. I saw a question earlier on that's now off my screen about what types of water bodies can you sample in, but maybe talk about the other mediums as well.
Yeah, so all kinds of water. So we've got samples from ponds, lakes, rivers, streams, estuaries, coasts, and open ocean, everywhere from the equator to the North Pole. But also, you can do the same kinds of things with soil and sediment. Mostly what you do, if you take soil and sediment samples, actually mostly what you're sampling is the organisms that are in those samples, rather than, you'll get some things that will leave bits of traces of DNA through the soil as well, but mostly it's stuff that's in there. Feces, so we do diet analysis and things, which we've done on bats and geese and otters so far. That works really well. You can do insect traps, an obvious one. There are some people who are starting to do air analysis, so microorganisms in the air. You can do pollen capture and sequence of pollen. Plants are much more difficult than animals for various reasons. They don't vary as much. The key genetic markers.
So Cheryl's asked, does it have a use in the construction industry through EIAs?
Yeah, it does. And I mean, I guess that's one of the examples where maybe there are some sort of barriers in terms of just there are very established ways of doing things with EIA. So if you get just a very mainstream regular EIA, people just follow quite a fixed formula, and so bringing something new into that is quite tricky. But when you've got EIAs that are in particularly vulnerable habitats, or you need to do a much sort of broader assessment or get an idea of just how things are changing, what your impact has been, and particularly how well you've done at restoring the habitat afterwards, whether you've managed to create a habitat that looks like the original one. Using invertebrates to kind of get a biodiversity fingerprint of the habitat is really powerful. And then obviously you can do, CAR can get involved with bits of EIA work when there are key species that need to be detected. So it's a good way of detecting things like eels.
Excellent. So I think you have some slides next about the kind of entrepreneurial side of things.
Yeah.
We could probably spend too long talking about the science.
We could. I wanted to just talk a bit about the challenges that I faced. I put this in black because I felt, probably for the first 2 years, I just felt like I was in the dark all the time because, I was setting up a business, but actually I didn't know anything about any of the components of a business. I hadn't even been employed in a business before. So, the basics about employing people and, what are the legal implications of everything you do, and what about IP, and what about finance and accounting and forecasting and marketing and websites and all of those things, let alone running a commercial laboratory and all of the lab management systems and the accreditations and all of those sorts of things. I think it's really true that you learn best from getting things wrong and then realizing how you should have done it. And that's how I've learned most things. It's really effective, but it's really uncomfortable. And you spend a lot of time feeling like, you're blindly trying to make everything up as you go along, at the same time as having to, project confidence to everybody that you know what you're doing.
Because there's always a trade-off in small businesses, isn't there? That you spend the time learning how to do something yourself, or you buy in that expertise. Expertise tends to be quite expensive, but also people aren't very good at attributing their own learning time to the company balance sheet. This is an actual expenditure because it's been my time. Did you feel that you got that trade-off right in the early days of setting up the organisation?
I think in the early days I probably took too much on myself, but that was a consequence of, we didn't raise a huge amount of money to start with, so I didn't have a lot of extra money, and we actually needed the people to run the lab. So the people I employed were the technical people who could do that side of it. So that was, the first thing that I guess I divested myself of, was the day-to-day sort of decisions on what to buy and what to do with these samples. I'm now getting to the point where we've outsourced HR and accounting and that side of things. I've got somebody who's helping me on the finances, who's made a huge difference. It's sort of getting there.
Yeah.
But I think one of the things I just wanted to point out really is that I think it can take a huge toll on your emotional health, because there's a huge amount of pressure on you really as an individual at that level, to be projecting this image of confidence, and your staff aren't going to work for you and do a great job and be motivated if you don't look like you believe in what you're doing, which you do deep down, but you have days when you doubt yourself, and your investors aren't going to continue backing you if you're not sort of showing them that side. And so it's difficult to know when you have those moments when you really doubt yourself or your ability to do this, and actually it's very difficult to know who you can talk to about it within your sort of company, to get that kind of support. And I do think that there's within the tech accelerators, there's a great sort of peer network of support. So whatever drama you're going through emotionally yourself, somebody else will either be going through it at the same time or went through it last week. Whereas when you're on your own, you don't have that.
So how do you tap into those networks of people in the same position as you?
So for me, I was lucky that coming back to the investors, and they had other companies that they'd invested in and other founders that they were able to link me to talk to, and I had some I still have contacts from my time back in Wira that I could tap into. And I just knew that there were those networks there, where I think it was very easy not to know that they were there. And I think that there's a bit of a lack of support out there for entrepreneurs. You're working really, really long hours and giving up everything to do this.
What I found very interesting from our first episode of this series was talking to Will Pope. He said that entrepreneurship is nearly always framed in the media as being about being an ideas person, being the innovator or the inventor. But what he was very keen to point out was that the real entrepreneurial skill was taking that idea and building it into a viable business. And I brought this up with the second episode as well with Joe, and he was at that stage where he was a bit concerned about, was he the right person to take it forward, because he felt that he was much more comfortable coming up with ideas. So I wonder if you could just reflect on that in your own personal experience about which part of entrepreneurship you prefer.
Yeah, so I guess I was an accidental entrepreneur. I never sort of set out being like, oh, I'm the sort of person who comes up with crazy ideas and I'm going to go and start a business. It was kind of one thing led to another. And when I look back, there are things in my past that I can see, okay, these traits are the ones that sort of led me to being where I am. But I've been through that exact process, actually, of thinking, you get to a point where you think there's so much that I'm supposed to know and so many balls I'm supposed to be juggling, and I could drop any ball at any moment and the whole thing can fall down and it would be on me because I don't know what I don't know. And you used to just think, oh, maybe maybe it would be better to bring in somebody who can actually sort of, grow the business and run the business and everything. But actually, I'm the one with it's not necessarily the creative spark, but with the real deep understanding of what we're doing and why and what we want to get out of it. And I think just coming back to that, probably at that time we got a little bit of external validation from just some of the ideas that I had about where this could go, just started to be gradually validated, and I was like, okay, I wasn't talking nonsense. Like there is something there. I did have some good ideas, and then just gradually building confidence, and now I'm really enjoying that process of learning and sort of stepping up and leading it. And I guess some of that is just having a bit more stability, a bit more sort of confidence and track record, and having already learned a lot. But
so outside of the passion for the subject and the understanding of the science, do you think that there's anything that someone with a scientific background brings to the business side of it, that someone who's gone through the traditional MBA route and is your kind of serial entrepreneur doesn't have as part of their skill set?
I don't want to be taken the wrong way, but I think there's a real honesty there with scientists. And I think it actually mostly plays against us, in that when you're going out there and raising money in particular, a lot of it is, it's not make-believe, but it's bordering on, make-believe. And you have to be able to sort of step out of reality and say, what if, everything went well, and people go, just show me some bigger numbers, and you go, okay, I'll show you some bigger numbers, and then they go, oh, those are too big, show me some smaller numbers, and you think, okay. And as a scientist, I found that really difficult to do. But on the flip side, it's made me very able to talk to my customers and clients and communicate very effectively with them and build up a level of sort of trust and understanding that has actually been really valuable. And I guess as scientists, we're trained to be analytical, we're numerate, we're trained to present things. So much of my life now is giving presentations and talks and things, and actually those are all skills that I first started developing as a scientist.
It may not be possible to categorize it with the number of customers that you have these days, but who generally in those organizations are making the purchasing decision from you? Is it someone in the finance department, or is it a scientist making
No, they're
decisions?
mostly scientists. So
Okay.
mostly we're talking to
So there's
ecologists.
probably that shared language with them as well.
Yeah, there's definitely a shared language. I mean, the kind of ecology that happens in the real world is quite different to the kind of ecology that happens in the academic world. So for me, you know, actually I've learned a huge amount about that as well through this process. Oh yeah, this slide just has some of the things that I printed on our wall when I was going through some moments of self-doubt. I love that quote from Winston Churchill: "When you're going through hell, keep going." And then the poem about what's it called? I can't remember what the poem's called, but I had it printed on a tea towel once. Success is failure turned inside out, silver tint in the clouds of doubt. And you never can tell how close you are. It might be near when it seems so far. So stick to the fight when you're hardest hit is when things seem worse that you must not quit. But I had lots of sort of pep talks myself.
So as much as the kind of entrepreneurial skills around knowing HR, knowing finance, being able to do kind of projections, what I'm hearing quite clearly from you is that the hardest part of the journey really was the emotional side of things, keeping up your spirits and.
Yeah, and that might be just me because I'm probably quite an emotional person.
Playing too much of that game my emotional intelligence.
Exactly, I'm far too emotionally intelligent. No, I mean I'm naturally sort of at the sort of empathetic and emotional end of things. So for me, that was sort of the big challenge. And yeah, I think it can be quite lonely, but it's worth sticking out. I really think it's worth sticking out.
So they often talk about this in books on entrepreneurship, about those first couple of years of starting a company and the kind of hellish burden that it puts on your kind of work-life balance and your social life. And I guess you're getting to the point where the payoff starts to happen.
I hope so. I would like to thank my husband for all of his support during the last couple of years, and also my rowing club, who get when nothing can come out at work or everything comes out at rowing. So they've borne the brunt of me. But yeah, I'm hoping to become a much more stable person over the next couple of years.
Excellent. I noticed when looking on your company website as well that you have a number of scientific advisors and non-exec directors. Can you talk about the support that maybe they've given you as well?
Yeah, so the scientific advisors are my co-founders. So Doug is a molecular ecologist, the professor at UEA, and also works in China. And Alfred is, really a molecular taxonomist, been at the forefront of the sort of technical side of things. So they provide sort of sounding boards and specific advice when we've got challenges on scientific sides of things. The non-exec directors are my board members, so they're investors and they're just the absolutely sort of crucial business advice and support. And then we've got advisors who sort of come from the world that we're trying to address, who can give us more advice on whether what we're putting out there is really needed, or a bit of a steer on direction and market and that kind of thing. Okay. So yeah, just quickly, I guess the commercial challenge that we faced was being in a new market where people didn't know that they wanted what we did. So how that process of building trust and we took a decision very early on that we weren't, we could have been very protective about everything and said, look, we do magic. Or you can go and sort of open it up and say, hey, here's what we're doing. Come in and understand it, and then we'll sort of show you how it works. And so we went down that much more open route to sort of build trust, and also to allow us to build up a network of collaborators across the research world, who have been amazing network of people. But one of the big challenges has been that you've got a new market and people sort of jump into it and see an opportunity, and you end up with people jumping in who don't have that kind of background in it, and they do sort of a quick and dirty analysis, it gets weird results, and people go, oh, we tried eDNA and it gave us weird results, and so we're not going to try that again, and sort of throw the baby out with the bathwater. So there's been a lot of just sort of trying to build industry trust. And then, yeah, finally, I just wanted to touch on, because I know it all sounded a bit negative, but the sort of really rewarding sides of doing this has been, you know, for me particularly, seeing my team start to just take everything on themselves. You know, this has got a picture here of my lab team sort of having a lab meeting and discussing how we were going to arrange our new space and our systems and our tracking, because we were taking on a new lab space. And you know, now I can just sort of leave them all to basically run the day-to-day stuff, and it feels like we've grown up a lot. And silly things like just kitting out our office with matching desks and looking like a proper little business, as opposed to sort of making do with a tiny space in a tiny room that crammed us all in. And then getting, you know, when you get feedback from your customers that sort of say, you know, this is amazing and it's really helped us, and we could never have got this sort of data otherwise, and you know, that's just a brilliant feeling.
Do you notice the nature of your role changing a lot then, in terms of the percentage of your week that you allocate to certain types of tasks?
Yeah, so to start with, it was, you know, I was involved in every single decision day to day. You know, should we buy this kind of tube or that type of tube? Should we do this sample or whatever? So gradually, I've sort of been able to step back from that and let my sort of head of science take that over, run his own team. So now I'm able to spend a lot of my time out and about, talking to people, going to conferences and meetings, and doing different sorts of things. And then I just finished off with some advice to anybody else sort of thinking about going down this route. One of the things that I was told at the beginning and didn't appreciate the importance of was having a full-time co-founder can, I think, make just a huge difference, because you've got somebody else sharing that burden with you on a sort of day-to-day basis, in the trenches and going through the ups and downs themselves. And I had, you know, my co-founders are amazing, and they the people whose work this was all founded on, but they weren't there on a day-to-day basis, so they couldn't do that for me. So I think have somebody else in there from the beginning. And ask for help when you need it from whoever you can. And I think the thing that I've really realised is that so much about running a business is about the relationships that you build, the relationships with your staff, the relationship with your investors, the relationships with your clients and other people in the field that you're working in, even your relationships with your competitors. You know, those are the things that sort of give you the ground to run on. So just be nice to people and build good relationships, and you get a long way.
Next week we have a report coming out on employability skills in environmental science degrees, the transferable skills. It's not too late for us to stick in a last minute addition. What is missing in an environmental science education as it's currently taught that you think might encourage people to be a bit more entrepreneurial with their approach to the science of environmental science?
Oh, that's a really good question. I mean, I think that in general, teaching at universities has been very, you know, it's very much kind of assumed that it's a linear process and that you're within an academic system, and the aim is to get on to the next academic rung. And I think just sort of, you know, awareness that there are all sorts of different things that you can do, but I think also the awareness of the people often think that, you know, I've learned all of this stuff, but it only qualifies me to do more of what I've done. And actually, it's not so much learning new skills, it's an awareness of really how transferable those skills are. So, you know, particularly things like being able to give a good, clear, concise presentation is an amazing skill, because it allows you to stand up and communicate what you do. You know, I don't think you can train people to have ideas, but you can train people to understand that the skills that they're developing are, you know, skills that will help them across any walk of life.
Okay, well, the hour has flown by. It's been fascinating to learn both about what your company does, your own history, and your thoughts on the topic of entrepreneurship. So, thank you, Kat.
Thanks for having me.
This topic is very much tied to kind of natural capital, and it's also this theme of this year's Burntwood lecture, which will be given by Kathy Willis of Kew Gardens on the 8th of November. I believe it's almost fully booked, so if anyone wants a ticket, they're going to have to hurry along to the IS website in about the next week, I should imagine. The next webinar we're running is a couple of days earlier than that, on the 6th of November, on the theme of the future of tropical agriculture, with Ed Turner of the University Museum of Zoology in Cambridge. That's part of a biodiversity week that we're having at the beginning of November, so sign up to that online too. Thanks very much.
Thank you.